This series of video discusses the histological aspects of cartilage and bone. The session objectives are listed on this slide and are also available in your lecture notes. Cartilage and bone has some common features. As such, they are both specialized connective tissues in which the cells are surrounded by extracellular matrix, or ECM. In these, the ECM volume is greater than the cells. ECM in these tissues is mostly collagen and glycosaminoglycans. The composition of the extracellular matrix is dependent on the tissue function, as it will become apparent later. Both cartilage and bone are of mesenchymal origin. So the ECM and the structure differences dictates the properties of the tissue. So let's talk about the cartilage first. So cartilage is a product of resident cells known as chondrocytes. The cartilage itself is rather unique in that it lacks blood vessels. So the blood supply to the tissue actually comes from outside of it, and the nutrient and metabolites are moved into the tissue by diffusion. It also lacks nerves. As such, the pain that's originated from within the joints, which you will see later, comes from the surrounding tissue and not the cartilage itself. The cartilage can be classified into three different types: hyaline cartilage, elastic cartilage, and fibrocartilage. This table from your recommended textbook summarizes all the features of these three different types. It is recommended that you review this material and look at the similarities and differences between the three types. It would help you to identify these three different types in histological sections. We would not talk about this table in details in this presentation, but it is available in this slide for your review. So first type of cartilage, the hyaline cartilage, anatomically can be found in the embryonic skeleton, and it also acts as a supporting structure in the trachea and the nose. It's also seen in the costochondral junctions in the ribs. But most importantly, it covers the articulating surfaces of joints, and that is where we will be focusing later on. Extracellular matrix of hyaline cartilage is composed primarily of type 2 collagen, but there are other minor collagens also present, and that includes type 6, type 9, type 10, and type 11. The other component within the extracellular matrix is the aggregans, which are proteoglycan molecules rich in chondroitin 4 sulfate, chondroitin 6 sulfate, and heparin sulfate. Some of this information is covered in biochemistry and will not be discussed here. Other components include non-collagenous proteins such as chondronectin, as well as multiple growth factors. This list of material listed here is not exhaustive, and it's just for information. If you were to look at the schematic of the extracellular matrix and the cells within the hyaline cartilage, you will notice that you have a large portion of that covered by the extracellular matrix, but you also see cells. So these are represented as these big areas of cells with nucleus in the middle, and they're usually seen as either a pair or even as multiple cells together. They originate from cell division and they appear as pairs, and this is a very characteristic hallmark of cartilage and should help you in identifying that the tissue that you're looking at is cartilage. The extracellular matrix itself, as you can see here, is shown to comprise of type 2 collagen here and proteoglycan monomers on the backbone of hyaline, which is forming the aggregans in total. So all in all, this is a schematic demonstrating what cartilage is composed of. Now, if you were to look at the histological section of that, you will see something like this. So here you see a large area of cellular population, as well as you have this outside dense connective tissue. But in between there is a very small thin layer of cells which are referred to as the perichondrium. The perichondrium is equivalent to a population of cells which eventually give rise to the chondrocytes in the cartilage. And as cartilage grows, these cells proliferate, as you can see in this lighter shaded area or lighter stained area here, which eventually proliferate. You can see the cells here dividing actively, and then they synthesize the extracellular matrix surrounding themselves, and that eventually becomes what is known typically as this hyaline cartilage. As I mentioned before, you can see this kind of clustering or pairing of cells, which is pretty much a giveaway for looking at what cartilage should be. So if you were to look at schematic of what's going on in the matrix and the cells themselves, this picture on the right shows you individual cells as they have divided into a cluster of 4 cells here. And you notice that although we are not seeing endoplasmic reticulum in histological section, but because they are producing a large amount of extracellular matrix, it's very active. The other thing that you notice is that it has these surrounding darker stains, and they are apparent here, for example, in the actual histological section, and that's referred to as the capsular matrix, and that kind of contains the cells within its lacunae. Moving further away in the matrix, you have this area referred to as the territorial matrix, and that is a little lighter staining than the capsular matrix, and you can see that here in this particular area or around the cells all over here. As we move away from that, you see some interterritorial matrix which appear as the lightest of the whole group, and you can see them right around here. In a histological section, you also notice there is a composition going from the surface to the deeper zone of articular cartilage. So this is a section taken from the joint, and you can see the articular cartilage going from the surface to the deep, and you refer to as a superficial, middle, and deep zones. So cartilage itself grows by 2 mechanisms. It can either grow from within, by which you saw earlier the cells were growing the matrix surrounding themselves and expanding the matrix, or it can grow by what is referred to as appositional growth, where the growth is happening on the surface, and that is where the original matrix is being expanded on by placement of a new matrix on top of that, and that tends to happen near the perichondrium. The other type of cartilage is the elastic cartilage, and it is seen in the ear as well as the auditory tubes, the epiglottis, and in the larynx. To be able to see the elastic cartilage, you have to stain it with special dyes which will stain the elastic fibers, and that dye, the Orsine Rigot dye, is used for revealing the elastic fibers. And this histological view, which you will be seeing in your labs, represents that. And here once again you can see these doublets of chondrocytes, and then the matrix in this case is appearing rather dark because all the elastic fibers within it are staining with this darker staining dye. So third type of cartilage was the fibrocartilage, and this is very similar to the hyaline cartilage, with one exception, that it has type 1 collagen in addition to the typical type 2 collagen. And but it also appears as more like a dense regular connective tissue than the hyaline cartilage, which has a more uniform appearance. Anatomically, you will expect to see fibrocartilage in the intervertebral discs, in the pubic symphysis, as well as where the tendons are inserted into the bone. So if you look at histologically, this is the kind of picture that you expect to see. As you can see, wherever you have the chondrocytes, they appear rounded cells, whereas fibroblasts are like typical elongated fibroblasts. And you can also see, because of type 1 presence, you can see that it has some kind of orientation which appears like a dense connective tissue.